Hello students, in this video lecture we are going to learn about the first step of gene expression, which is called as a transcription, right? So in transcription process what happens, whatever the DNA which is present inside the nucleus, right? So that DNA undergoes the process of transcription and thus form the structure of RNAs, right? So during the transcription process, okay, so the DNA, it is get transcribed to form the mRNA, right? And this mRNA again get undergoes the process of translation and forms the protein. So this is the central dogma of cell, right? So in this process we are going to learn about the transcription process, how DNA get converted into the RNA. So that we are going to learn in this lecture video. So first we'll start with the definition of transcription. So transcription, it's a cellular process in which the RNA is synthesized using the DNA as a template. Such process is called as a transcription, right? So here you can see the structure of this DNA, which is double stranded. So this DNA will convert into the RNA. So DNA may be double or single stranded, but it will be converted into the RNAs, right? So such process is called transcription process. But most of the living organism, they have the double stranded DNA, like the higher living plants as well as animals, right? We humans also have the double stranded DNA, right? So such kind of double stranded DNA get converted into the RNA, right? Single stranded RNA. So such process is called transcription process, right? So this is the first step of gene expression in the is called the transcription. And the RNA polymerase, it's an enzyme, right? As like the DNA polymerase we have learned in the DNA replication process. So this RNA polymerase play important or major role in the transcription process, right? So three different types of RNA molecules are synthesized during the transcription process, which are namely mRNA, right, ribosomal RNA and transfer RNA, right? We will shortly, it will call as a mRNA, rRNAs and tRNA. So in the prokaryotic system, in the prokaryotic cell, the single RNA polymerase is perform the process of transcriptions, right? So it transcribes all the three different kinds of RNAs in the prokaryotic system, whereas in the eukaryotic system, in the eukaryotic cell, three different RNA polymerase, namely polymerase first, that is RNA polymerase first, RNA polymerase second and RNA polymerase third. They are involved in the synthesis of three different RNA molecules, that is mRNA, rRNA and tRNA, right? So the process of this transcription, it involves the three different steps that we'll see here. So first we'll see the RNA. So RNA is a polymer of ribonucleotide held together by the 3' to 3' to 5' phosphodiester bridge and are single stranded, right? So it is the only molecules known to function both in the storage as well as the transmission of genetic information and in the catalysis also. So all the RNAs except the RNA genomes of certain viruses derived from information which is stored permanently in the DNA. It means all the living organism, right, which is having DNA as a genetic material, they have stored the information, right, which is permanently in the DNA. So that will be get transcribed to form the structure of RNA, except the living organism like the COVID virus, coronavirus, right? So it's having the RNA as a genetic material, and different kinds of another viruses, that is retroviruses, they are also having RNA as a genetic material. So that RNA is directly get translated to synthesize the proteins. But if the COVID virus, it will infect the human cells, then it will inject its RNA and that RNA will again convert into the DNA. So that is called as a reverse transcription process. And during the reverse transcription process, RNA will convert into DNA and again DNA will convert into the RNA and further into the proteins. right? So in this way the retrovirus perform their life cycles. The RNA, there are three major kinds of RNAs. mRNA, right, which is which varies from 5 to 10%, which transfer the information of genes to the ribosomes, right? You know the ribosome. So that is the which are involved in the protein synthesis, right? In prokaryotic as well as in the eukaryotic system also. Then tRNAs. So these tRNAs are also 10 to 20%. So they read the codes on the mRNA and transfer appropriate amino acids to the mRNA. And the third kind of RNA, which is having the high concentration in the cell, that is called as a ribosomal RNA, right? Which are 60 to 80% of total RNAs. The These rRNAs is the constituents of the ribosomes, right? So many additional specialized RNAs which has the catalytic activity or the regulatory functions, they are present in the cell. So here you can see from the DNA you can synthesize the RNAs, mRNAs, tRNAs, okay, then miRNAs, okay, that is micro RNAs, then ribozymes, U1 RNAs, which are called as spliceosomes, right? So from this DNA different kinds of RNA can synthesize, right? In replication, the entire DNA molecule is normally copied, right? And in the transcription, the particular gene or the group of genes which are copied at any time and some portion of the DNA are never get transcribed. That is called as a euchromatic region and heterochromatin region of the DNA. So the genes, which is a segment of the DNA that codes for a type of protein or for RNA, and may present on any strands of DNA which contains many genes. So here you can see the structure of this DNA, and there is a specific region of the DNA which codes for the protein, that is called as a gene, right? And remaining portion of the DNA, it is non-coding region, means it's not going to convert into the RNAs. Differences between the replication and transcription process. In the previous lecture we have seen the replication process and transcription process. So in the replication and transcription there is a difference in the template. So in the replication both the strands, okay, they are involved, and while in the transcription only single strand of DNA is involved for the synthesis. And here in the replication process whole genome will be get replicate, right? While in the transcription process small portion of genome get transcribed. In the replication process the primers, they are necessary, while in the transcription they are not necessary. The enzyme which is involved in the replication process, DNA replication process, is called as a DNA polymerase enzyme, and enzyme which is involved in transcription process, it's a RNA polymerase enzyme. So product of this DNA replication is the double stranded DNA, while the product of transcription is the single stranded RNA. So very important point is there, you have to focus here. In the DNA having adenine, thymine, guanine and cytosine, means adenine will pair with thymine, guanine, and guanine will pair with the cytosine. But in the transcription process adenine will pair with the uracil, and if the DNA having thymine, then it will pair with the adenine, okay. So it means the thymine is missing in the RNA structure. In case of that uracil is there, and guanine will pair with the cytosine. So proofreading activity is there in the DNA replication process, while absent in the transcription process. Then we'll see the features of transcriptions. So transcription is a highly selective process which is selectively due to the single embedded in the nucleotide sequences of DNA. right? So as it is a very specific process or selective process, so it sequences the marks the beginning and the end of the DNA segments which is to be going to be transcribed from. It means there is a two different points on the DNA from where the transcription will start and it will going to stop, right? So you will get the start point and stop point on the DNA sequences, right? Then many of the RNA transcripts, they are synthesized as a precursor, okay, that is known as a primary transcript, which is on the modification and trimming convert into the functional RNAs, okay. The site of the transcription in the prokaryotes is in the cytoplasm, while in the eukaryotic system transcription will be takes place inside the nucleus. And if the cell there is a mitochondria, then mitochondria will transcribe its own genome, means it will be takes place in the mitochondria also. If there will be a chloroplast, then inside the chloroplast also transcription will be takes place. The on the nucleolus ribosomal RNA will synthesize, and the nucleoplasm tRNA and mRNA will synthesize, right? So these are the transcription site of prokaryotes and eukaryotic systems. So the basic biochemistry of RNA synthesis is similar in the prokaryotic and the eukaryotic system also, but its regulation is more complex in the eukaryotic system, because eukaryotic system they are having their DNA which is having coding region as well as non-coding region. So that non-coding region will be going to be removed, and thus it will convert it into the coding region, that is the protein structure. It will code for the protein structure. So that's why it's complicated. So RNA synthesis in the prokaryote is catalyzed by the large enzyme that is called as a DNA dependent RNA polymerase, or RNA polymerase simply. A single enzyme, that is RNA polymerase, synthesizes all type of cellular RNAs in the prokaryotic system, while in the eukaryotic system specific RNA polymerase is for the specific kind of RNA, right? Excuse. RNA polymerase use one of the DNA strands as a template on which the complementary ribonucleotides they are incorporated into the synthesis of RNAs, right? So here you can see the strands of DNA which is going to be transcribed to the RNAs, that is called as a template strands, right? So opposite strands is referring as a coding strand, okay, and another strand will be called as a template strands. It means the coding strand will not convert into the RNA, only the non-coding strand, that is the template or antisense strand, so it will convert into the structure of RNAs. So here you can see the sequences. So 3 prime to 5 prime, see the 3 prime to 5 prime strands of DNA, right? It's a template or antisense strand, so this will convert into the structure of RNA. So cytosine, obviously it will convert into guanine, and adenine convert into the uracil, guanine convert into the cytosine, thymine will obviously convert into the adenine, thymine again convert into the adenine, adenine will convert into the uracil, because in the structure of the RNAs thymine is missing. In case of that, uracil will be there. So in this way this RNA structure will be get formed. Then we will see the RNA polymerase, right? So the E. coli RNA polymerase is one of the largest enzyme inside this cell, right? So it is composed of a core which is contain the basic transcription machinery and the sigma factors, right? So this RNA polymerase synthesizes the RNA in the direction of 5 prime to 3 prime. So that means the DNA template is read in 3' to 5' direction. The ribonucleotides, they require ATP, GTP, CTP and UTPs. Okay, so the prokaryotic RNA polymerase is a multimeric enzyme. Multimeric means multi subunit is there, which consisting of 6 subunits. Twos are identical alpha subunits, okay, similar but not identical beta and beta dash, and another one omega sixth, and sixth one is the sigma factor, right? So 2 alpha, beta, beta dash and omega will consist of the core enzyme, and when the sigma factor will be attached to it, then it will convert into the hollow enzyme. So remember that RNA polymerase is a hollow enzyme. Clear about this? Okay, so the core enzyme has this different kinds of subunit, okay, which consist of hollow enzyme structure. So for the initiation of transcription, the RNA polymerase bind to a specific site on the DNA, that is called as a promoter sequence. So this sigma factors allows the initiation of transcription by causing the hollow enzyme, right? The hollow enzyme means this RNA polymerase enzyme to bind tightly to the specific promoter. So the core enzyme has an affinity for the binding to the DNA non-specifically. Specifically means it will attach somewhere, but the specificity of this RNA polymerase enzyme is due to the subunit of RNA polymerase, that is a sigma subunit, right? So due to this sigma factor, the specificity to attach the RNA polymerase at the only promoter site of DNA is because of this sigma factor, right? So only sigma factor can therefore select which gene will going to be transcribed. Okay, so after the sigma has participated in the initiation, it leaves the core to carry on with the elongation process, right? So here you have seen the different subunits and what are their functions. So 2 alpha subunit, they determine the DNA which is to be transcribed. Beta subunit, it catalyze the polymerization reaction. Beta dash subunit, it bind and open the DNA template. It means it has the capacity to get unwind the DNA, as DNA is a double stranded, so it will convert into the single stranded and another single strand, that is called as the template strand. So that template strand will going to be transcribed. Then omega, so this omega function is unknown. Okay, up till now there is no any confirmed information regarding this subunit, that is omega subunit. And the sigma, it recognizes the initiation site, that is called as a promoter site. Okay, function of this RNAP, that is RNA polymerase, it's a short form of RNA polymerase. So a single RNA polymerase perform the multiple functions in the transcription process. So first, it search and bind to the promoter site. Second, it unwind a short stretch of DNA, double helical structure of DNA. Third, it selects the correct ribonucleotides and catalyzes the formation of phosphodiester bonds. That is called as a polymerization according to the best pair rules. Means adenine will pair with thymine, uracil will pair with adenine, guanine will pair with cytosine will pair with guanine simultaneously, right? So the combination of RNA with the NTPs will convert into RNA plus inorganic phosphate. Okay, so and then detect the termination signal. So this is the biochemical reaction, and this reaction will going to stop somewhere, right? So that stop point is called as a termination point, and this RNA polymerase detects that termination signal. Next function, that is the interact with the activator and the repressor, the proteins that regulate the rate of transcription. So transcription in the prokaryotes, there are divided into three different stages: initiation, elongation, and termination, as like the DNA replication. Okay, so initiation involves the RNA polymerase which recognize the promoter region and start the transcription process. So that is called as the initiation. Okay, elongation phase involves the RNA strand which is going to be continuously growing, and third and last stage, that is the termination phase, where the RNA polymerase stops the synthesis and the nascent RNA is separated from the DNA template, and in this way the termination of uh, transcription process will be takes place in the prokaryotic system. So first we will see the initiation of the transcription. Okay, in the prokaryotic system, I am talking about the prokaryotic system here, right? So RNA polymerase, it's a core enzyme which bind non-specifically to the DNA, right, which is refers to as a loose binding. So when the sigma factor binds to the core enzyme, so uh, it's form the hollow enzyme, and it markedly reduce the affinity for the specific site of DNA and enhancing the binding to the correct promoter binding sites. So for the antisense strands to become accessible for the base pairing, the double the DNA duplex, that is double standard DNA. So it must be unwound by the polymerase, right? So this property having by the beta dash subunit, right? So the initial unwinding of the DNA, it results in the formation of open complex, as like the DNA replication, right? So the sequences of the DNA, they needs for RNA polymerase to bind to the template and accomplish the initiation reaction which defines the promoter, right? So promoter are the characteristic sequences of DNA that is direct the RNA polymerase to initiate the transcription process. Okay, so usually it is located on the coding strands. Okay, the simplest type of promoter is found in the prokaryotic systems. So there are two general types of sequence elements which are found in the prokaryotic system. One sequence element is believed to be the promote initial binding of the enzyme RNA polymerase. Other elements usually has the high content of adenine and thymine. These sequences are like the 6 to 8 nucleotide in length and located about -35 and -10 base pair upstream of the start point of transcription. So from where the transcription will be started, it will be considered as a 0 point. Okay, and upstream to it will be negative, and downstream to it will be positive. Okay, so these are located at the -35 and -10 base pair. So these are non-coding strands which indicates the duplex DNA which is required for the transcription process, and change in only one base pair in the promoter region will decrease the rate of transpiration. Okay, so you can see here, so this is the start point. You can see here you have to be consider it as a 0, and upstream to it means at your left side. Okay, it will be considered as a upstream, and it will be denoted by negative sign, and at the right side of the start point it will be considered as a downstream, so it is denoted by positive sign. Okay, so you can see here the coding stands, right, which is coding and the template stand. So here you can see the sequences, the promoter sequences, they are actually on the coding stands, right? So you can see here Tata box, that is also called as a Pribno box. Okay, so from where the RNA polymerase will going to bound. So this is about the eukaryotic system. Okay, then we will see the actual initiation process. So here you can see the RNA polymerase, which is uh, denoted as a RNAP. RNAP stands for polymerase, remember, and this RNA polymerase having this sigma factor, you can see here, which is denoted by red color, So this sigma factor will come at the start point, okay, and it will bind to that start point, that is called as a promoter region, okay. And as this enzyme, that is RNA polymerase enzyme, having the beta dash subunit, so because of that beta dash subunit, it will break the hydrogen bond in between the two different strands of DNA, okay, and thus it forms the open complex. So here you can see in this slide, you can see the open complex. So here you can see the first nucleotide, it will going to be synthesized here, okay. Sigma factor is there, it will move towards the uh, downstream region, okay. So when the initiation will succeed, the enzyme releases the sigma factor, okay, and forms a ternary complex of RNA polymerase DNA, RNA polymerase with the DNA, and newly synthesized, that is the nascent RNA chain, will going to form, okay. So the polymerase then steadily progresses along the DNA, which allowing the reinitiation of transcription, okay, from the promoter by another RNA polymerase holoenzyme. So the enzyme, it takes around 1 to 2 seconds to clear the promoter, okay. So the region of unwound, okay, means the two different strands will going to separate, the two different strands of RNA will going to separate, right. So that region is called as a region of unwound DNA, that is called as a transcription bubble. It appears to move along the DNA with the polymerase, and the size of the transcription bubble remains constant at around 17 base pairs in the prokaryotic system. So the growing RNA chain, it forms a hybrid of about 12 bases with the template DNA strands, and the enzyme will move at an average rate of 40 nucleotides per minute. But however, this can vary depending on the local DNA sequences, okay. So to remain the transcription bubble, the RNA is unwound ahead of the bubble and is wound again behind the bubble, as like the zip, okay. So in this way the elongation will be start. So here you can see the continuous RNA strands will going to be formed here. So during the elongation, the enzyme would continuously go on adding the ribonucleotides, right, to the growing RNA chain in a template dependent manner, okay. So alpha subunit, it codes by the gene, that is RPOA gene, which reverse to assemble the RNA polymerase holoenzyme. So it has two distinct domains, N terminal domain and C terminal domains, so which fold indefinitely and are uh, tethered together by the flexible linkers, okay. So the N terminal domains is involved in the assembly of the holoenzymes, whereas the C terminal domains, it recognize and bind to up elements in the promoter stimulating transcription, and the beta subunit, it coded by the gene, that is RPOB gene, which binds to the nucleotides at the active site of RNA polymerase, where the phosphodiester bonds are formed. So this subunit is the catalytic center of the RNA polymerase, okay. So you can see here the transcription bubble, okay, it is get formed, and you can see how larger is the RNA polymerase. So in this way it is going to be moving forward, okay, at the downstream region, and unwinding as well as rewinding will be takes place, okay. You can see here the transcription bubble size also, which is varies from 16 to 17 nucleotide, okay, in the prokaryotic system. Then finally the process have to be terminate somewhere, because it's a what we can say, it's a biochemical reaction. So all the reaction have to be stopped somewhere. Okay, so when the polymerase reaches to the terminator at the end of the template, it fall off. Okay, and the template terminating the transcription. So two kinds of terminators are identified in the E. coli system, in the prokaryotic system. One is called as the intrinsic terminator, which functions without any help of from the others proteins, whereas the other is called as a row dependent terminator, which requiring the factor that is called as a row for the termination of transcription. Okay, so transcription region of the DNA template, it contains the stop signal. So prokaryotes have this two different kinds of termination process. First we will see the row dependent termination. Okay, so the signaled by a sequence in the template strands of the DNA molecule, which have such kind of the sequences, so which are 40 base pair in the length and are inverted repeats or hyphenated. So these signals they recognize by the termination proteins, that is called as a row factors, right? So that's why it is called as a row dependent termination. So row is an ATP dependent RNA stimulated helicase. It means it requires the ATP for the its activity, its functions, and it is depends on the RNA. So RNA will stimulate and thus it act as a helicase, so which bind to the signal, and thus the RNA polymerase cannot move further. So it is dissociated from the DNA that disrupt the nascent RNA DNA complex, which release the nascent RNA. Okay, so here you can see the purple color sequences there, which is the termination point. Okay, and rose factor will identify the stop signal, and the they shows the helicase activity. They separate the DNA from the RNA, and RNA nascent chain will go into form. Okay, another kind of the termination of transcription is row independent, right? So in this, the most independent terminators have two distinguishing features. One is palindromic guanine cytosine rich sequences, okay, which is followed by adenine thymine rich regions. So thus the RNA transcription of this palindrome is self complementary sequences, which permitting the formation of hairpin like structure. So I will show you how is the structure of that. So the second feature is highly conserved string of the adenine residues in the template strands, so that are transcribed into the uracil residues. Okay, so the RNA transcript the ends within or just after them. Okay, and you can see here the hairpin like structure. So this hairpin like structure will be formed at the palindromic sequences. Palindromic means the you can read the DNA sequences. They are exactly opposite to each other, like the adenine, guanine, cytosine, guanine, cytosine, and exactly opposite to that. Okay, guanine, guanine, cytosine, thymine. So these are called as palindromic sequences. And next to that you will find there is a region, okay, which is rich in the thymine adenine rich region, right? So there is a repetitive sequences, and thus the hairpin like structure will be formed, which is rich in the guanine and cytosine. Okay, and in this way the RNA transcription will going to stop. Okay, then we see the transcription in the eukaryotic systems. So it's much more complicated. There are different RNA polymerases which requires many transcription factor proteins, right? So transcription initiation, they need the promoter and upstream regulatory regions. There is a enhancer as well as the silencer also, right? So these are the DNA sequences that regulates the rate of initiation of transcription by RNA polymerase second. So there are three different kinds of RNA polymerase of eukaryotes. RNA polymerase first, second, and third. Right? And the product of this RNA polymerase 1 is the rRNAs. So that is 20S RNA, 18S RNA, and 5.8S RNA. Product of RNA polymerase second is the mRNA, right, or snRNAs, and third is responsible for the formation of tRNAs. Okay, and there is a inhibitor of this polymerase, that is the amanitin. It's antibiotics, and it is sensible for the um, RNA polymerase second only, and for third it will be a moderate. Okay, you can see here the uh, eukaryote promoters, which is called as a uh, Hogness box. There is a Tata box. Okay, from where the transcription will going to start. There is a cat box also, and upstream to that there is a enhancer or silencer region, from where the regulation sorry the transcription process will going to enhance, and there is a regulatory elements. Okay, so in this way the structure of this gene in the eukaryotic cells is get arranged as like here, like the regulatory elements, enhancer region, cat box, Tata box, and the transcription unit. Okay, so the sequences which is occupied by this cat box and the Tata box, which is near about 40 base pair, and remaining process like the initiation, elongation, and the termination, it will remain same as like the prokaryotic cells, right? So today we'll stop here. Thank you.